Good afternoon, ladies and gentlemen. I am Sujong Hong from South Korea. I really appreciate for the invitation of the chair for this plenary session. First of all, thank you very much for attending the 2021 APAACI Virtual Congress. I welcome you all APAACI friends to this session. I am very honored to host worldwide famous three speakers for this plenary session on the theme of microbiota and the immune system in health and disease. I will introduce all three speakers simultaneously right now. Then you can listen to the three consecutive recorded lectures. Then we will have time to Q&A for 10 minutes. The first speaker is Heidi Kwong. She is working for National Institute of Arthritis and Musculoskeletal and Skin Disease, USA. The talk title is Skin Microbiota in Health and Disease. The second speaker is Professor Harald Renz. He is working for Philipps University Marburg, Germany. Title is Microbiota in Early Life. The final speaker is Jenny Ting. She is working for University of North Carolina, USA. Her title is Microbiome and Innate Immunity. If you have any questions, please unmute your questions. We wish to extend and share our knowledge to all audience. I hope that we can enjoy this plenary session together. Now, let's start.
Hello, everybody at the Asian Pacific International Conference. My name is Harald Renz from the Philipps University in Marburg, and I really have to apologize that I cannot be face-to-face with all of you due to this very special circumstances with the pandemic. But I really hope that we have a fruitful and lively exchange, scientific exchange and personal exchange in the future. So today I have been asked to talk about the role and contribution of the microbiome for allergy and asthma development, and with particular focus on bacteria and viruses. And at the end, I will also discuss a few issues with the SARS-CoV-2 virus, of course. These are my conflicts of interest. And this is already the first important information I would like to transport today. And that is, when we are talking about the microbiome, we are not talking about a single individual microbiome at each person, but everybody of us is carrying a number of different microbiomes. On the right-hand side, you see the microbiomes divided into the 6 major phyla, from the gastrointestinal tract, starting in the oral cavity and going down to the colon. And it's quite apparent that the composition of the microbiome, even in the gastrointestinal tract, heavily depends on the anatomical side. And something very similar can be observed for the skin. And here, especially attributed is the vaginal microbiome, which not only consists of lactobacilli, but also of quite a number of other strains and colonies, which we will discuss in the next topic. The interaction with the microbiome starts already before birth. Already in utero, there is a very close interaction between maternal microbes and the fetal immune and metabolic system. Not only can these microbes are detected in the amniotic fluid, but the fetus swallows the amniotic fluid. One can detect actually intact, not live, but intact bacteria in the fetal gastrointestinal tract. And it is now very nicely delineated that these microbes contribute to immune development and immune maturation already in the fetus. And this continues with the next important event. And that important event is the question, how is delivery being performed? Are the babies born by vaginal delivery or by elective Caesarean section? And as you all know, the frequency, the prevalence of elective Caesarean section is dramatically increasing around the world. In Germany, we have now a prevalence rate of about 35%. And in some Asian countries, it is even higher. And it is now very well established that Caesarean section contributes as a risk factor to the development of non-communicable diseases later on in life, not only to food allergy and asthma and eczema, but also to autoimmune disease, to cardiovascular disease, and also to psychiatric disease. So the delivery at birth has an impact throughout life on the risk for the development of non-communicable diseases. And one of the factors which contributes here is the microbiome. Babies born by Caesarean section have a completely different initial seeding in the gastrointestinal tract as compared to vaginally delivered babies. Vaginally delivered babies are colonized by microbes from the vaginal tract of the mother, whereas Caesarean section-born babies are colonized mainly by skin microbes from the mother. And as we have seen before, the pattern of these microbes are completely different. So these are a number of factors and environmental and lifestyle conditions which contribute to the plasticity, to the dynamics of a microbial development within the first 3 to 4 years in life. And this slide should indicate that it takes some time until a stable colonization pattern has been achieved. And this time period is the most critical window of opportunity and also window of high vulnerability, where virtually all different environmental factors the baby is exposed to have an immediate impact on microbial composition. We have already talked about a little bit about the mode of delivery. Breastfeeding is another factor. The maternal communities on the skin, the oral and the gut communities contribute to neonatal and early childhood microbiome. The question of introduction of solid foods and of course antibiotic treatment. Antibiotic treatment courses, the number of antibiotic treatment courses in early life are also important contributors here. So it is very well established that dysbiosis at various body habitats has been firmly linked with many chronic inflammatory diseases, including asthma, eczema, food allergy, polyposis nasi, and also COPD. And this dysbiosis can be detected in different body habitats at different sites. But most importantly, dysbiosis usually precedes the development of the disease. So dysbiosis is present before the disease, before the inflammation really occurs. And this relationship between dysbiosis and disease development has led to the hypothesis that the microbiome actually causally contributes to the development of chronic inflammatory diseases, including allergies and asthma. And many, many factors, particularly in the postnatal life, can be attributed as risk factors for the development of the diseases and risk factors which have an impact on the microbiome. The microbiome is the intermediate between the environment and the immune system and the metabolic functions. And when we are looking on a global level, we observe that mainly urbanization, industrialization, and westernized lifestyle are a major driver for changes in the microbiome and subsequent development of the diseases. And this has been summarized here in a recently published review article from our group. And as you can see here, many of these lifestyle factors are dependent also on each other and build a highly complex network of factors which are in relation to each other. So a major cause for the increase in non-communicable diseases we are observing not only in the northern hemisphere but also on the southern hemisphere of the globe are industrialization and urbanization. Urbanization with traffic, pollution, stress, and more indoor than outdoor activities, the reduction of major infectious disease due to increased hygiene measures. We see that now with our masks. Changes in the diet with the westernized diet, high caloric intake, high intake of saturated fatty acids and carbohydrates. All of this contributes to changes in the microbiome, the diversity of the microbiome, and the loss of diversity, reduced diversity is actually attributed to the loss of protective so-called old friends. And we need to better understand these old friends, how they actually contribute to diversity and how diversity contribute to health. So loss of biodiversity is the global pattern, which is also actually linked to climate changes as well. And when we break this a little bit down, here the example of asthma prevalences around the world, we can actually distinguish 3 different patterns. The northern hemisphere pattern with a marked increase in asthma and other non-communicable diseases, particularly occurring since the end of the Second World War, and now probably reaching somehow a plateau. The intermediate pattern in developing countries like Russia and China, and the blue pattern, the most recent dramatic increase in NCDs, including asthma, occurring in sub-Saharan Africa. Here in Africa, we can now observe a change in diseases, in disease pattern, in disease occurrence, which happens in a time frame of about a decade, and 1 decade in Africa reflects 2 generations in the northern hemisphere in order to see these developmental changes. And these developmental changes are mainly, not exclusively of course, but mainly related to this increase in obesity. And you see here in red and in the orange and yellow colors the situation in Asia, in the Asian Pacific region where we see the most dramatic increase in obesity in girls and in boys on the right-hand side. And this pattern is related, obesity is related and to other comorbidities such as cardiovascular disease, hypertension, metabolic disorganization, and type 2 diabetes. And these changes in the diet have a marked impact on the microbiome. And as I said before, the microbiome is the intermediate for the development of the diseases. And this has been nicely delineated in many, many mouse models. There is only one mouse model in this regard, where mice were fed such a Western diet and they became obese. And now one can actually transfer the phenotype obesity in this case by transferring the microbiome from the obese mice to the lean mice. In this regard, one can not only transfer this metabolic phenotype, but also one can transfer the development of food allergy and the susceptibility to the development of food allergy. So these data nicely put together a mechanistic frame, bringing the microbiome in the center, in the focus between lifestyle conditions, dietary conditions on the one hand side, and inflammatory patterns on the other side. Now, over the time, we have learned a lot about pro-tolerogenic and anti-inflammatory bacteria. And on this table, I have summarized just a few of these good guys, if you want to say. This includes segmental filamentous bacteria, Bacteroides, Tetraiota micron, Clostridium clusters, Eubacteria, Butyrococcus, and Lactobacilli species, among others. And what is particularly noteworthy here is that we know important mechanisms of how these microbes contribute to the development of tolerogenic immune responses. And one important aspect in this regard is the production of so-called short-chain fatty acids. So the diet is, in the case of short-chain fatty acids, an important and relevant contributor for the production of these short-chain fatty acids by certain microbes. And these microbes are fed by non-digestible carbohydrates. Fibers are non-digestible for us human beings, but they are the excellent food source for certain microbes. And these microbes make short-chain fatty acids out of these non-digestible fibers. And these short-chain fatty acids contribute to the development of regulatory T cells, to the integrity of the epithelial barrier, and they have anti-inflammatory properties. So this is only one of many examples of how dietary and environmental components have an immediate impact on microbial colonization, on microbial products, and the host homeostasis in terms of inflammatory and metabolic parameters. And some of these environmental factors are good, like here in blue, and others are not so good, shown here in red. So there's a good side and there's a bad side of this diet microbe immune modulation. Coming back to the important role of short-chain fatty acids, what you see here is the result of one result of the important Pasteur cohort study, which is a prospective longitudinal study on the development of asthma and allergies in early childhood. And what the investigators could demonstrate here is a very firm link between diet in early life, fiber-rich, the production of short-chain fatty acids, butyrate and propionate, the development of regulatory T cells in the babies, and at the age of 6 years, a reduced risk for the development of atopic sensitization, asthma, and food allergy. As I said, one of several examples of how microbes actually impact immune and metabolic functions. So when we look into the good side and the bad side of microbes in terms of asthma or allergy development, we have identified over the years a number of microbes which carry an increased risk for asthma development. And this includes Streptococcus, Haemophilus, Moraxella, Klebsiella, Neisseria species, particularly colonizing the upper respiratory tract, the oropharyngeal space. This is now very well established, although the mechanisms of how particularly these microbes carry this increased risk to the young infants and also to the adults is not really yet delineated. But this is not restricted to bacterial communities. Of course, also viruses, certain viruses, respiratory viruses, carry a risk for the development of asthma and asthma exacerbations, and this includes the influenza virus, the rhinovirus, and RSV. And on the other side, we have learned from environmental studies that there are, of course, also certain protective microbes in the environment which help actually to train the immune system if we have a chronic and long-lasting exposure to natural sources of biodiversity from the communities. And one of the most interesting candidates in this regard is Acinetobacter lophii. Acinetobacter lophii has been actually identified in traditional farm environments originally, but there are now quite a number of exposure studies in Europe and beyond Europe identifying this microbe as one of many protective bacteria. So as I mentioned before, biodiversity is the overarching concept these days. Biodiversity is the enhanced exposure to biological materials, to biological microbes, microbial load, and to diversity. And biodiversity has been shown originally in the agriculture and farming studies, comparing industrialized and traditional farming. In anthroposophic lifestyle studies, biodiversity is also important in terms of consuming unpasteurized milk, pets at home, the high frequency of upper respiratory tract infections, the birth order, the daycare issue, all of this contributes to biodiversity and has resulted, I call it, the inverse hygiene hypothesis. Inverse because here we are talking about broad exposures to biomaterial as a good thing in life. And this is the example of traditional farming and early immunoeducation. And I would like to emphasize that on the mechanistic level, one important mechanisms of how microbes actually interact with the host's immune system and with the host metabolic function is via epigenetic regulation. And there are many, many different machineries of epigenetic regulation. DNA methylation or histone acetylation are only two of several of these machineries which the cells have available in order to biochemically modulate the accessibility of transcription factors to the respective genes. We have been very interested in analyzing the contribution of epigenetics in linking environment, nutrition, and the microbiome. And here you see some overview articles recently appearing from our group in order if you want to read a little bit more about these machineries. So one of the aspects I mentioned already is the exposure to short-chain fatty acids produced by gut microbiome. Now we have been able to show that these short-chain fatty acids induce Th1 factors in CD4 T cells through changes in histone acetylation patterns, and that they promote iTreg development from cord blood or adult peripheral blood CD4 T cells, again through histone acetylation of several important genes. Further on, also this farm-derived gram-negative bacterium Acinetobacter lophii protects through epigenetic mechanisms, again in this case changes in histone acetylation, particularly of the interferon gamma gene and the interleukin 4 gene, leading to a downregulation of Th2 phenotypes and increasing the capacity to make regulatory T cells and Th1 immune responses. So not only Acinetobacter lophii is working through epigenetic regulations, but also other bacteria and even nematodes are operating through epigenetic mechanisms in order to change the Th2 inflammatory pattern. In the case of chronic schistosomiasis during pregnancy, we see a downregulation of Th2 phenotypes through histone acetylation pattern, particularly in the interleukin 4 and the interleukin 5 genes, whereas nematodes tend to change the epigenetic profile in a way that we see an increased and elevated and augmented Th2 immune response. So these data indicate that the link between the genetics and the environment in terms of the development of the clinical phenotype is really working on the level, on the molecular level of epigenetic regulations. And microbes contribute to this exposome very importantly, because many lifestyle conditions like urbanization, industrialization, Western diet have a very, very strong impact on microbial composition and microbial activities. So we have addressed the issue of risky bacterial exposures, the issue of viruses which carry the risk for asthma exacerbation. And I have shown you a couple of examples of protective environmental microbes, how they impact actually the host's immune response. So now for the last part of my talk, I would like to ask the question, how about SARS-CoV-2? We are in the midst of a pandemic. Has the SARS-CoV-2 virus also an impact on allergy and asthma? I think this is a very important question, and I would like to give you a few answers from our studies, from our recent studies here in Marburg and with our collaborators. So we have learned throughout the pandemic that the COVID-19 disease, of course, it starts in the upper respiratory tracts, goes down to the lung, but then at the very end, eventually COVID-19 disease can be considered a systemic disease affecting many different organs, the blood system, the immune system, and the clotting system. So it's a chronic inflammatory condition caused by the SARS-CoV-2 virus. And we had the opportunity in early 2020 to analyze with our friends and collaborators at the Tongji University in Wuhan in China, the first 500 inpatients divided into severe corona and non-severe corona causes. And of course, when we look into the severe corona causes, it became evident at that time that many comorbidities can be considered risk factors for severe COVID-19, including diabetes, hypertension, coronary heart disease, chronic kidney disease. But when we looked into asthma, we observed something very peculiar. Although asthma is a chronic inflammatory lung disease, the prevalence of asthma among severe COVID-19 patients was very, very low in Wuhan. And this triggered further research in our group, and we investigated the prevalence of asthma among COVID-19, hospitalized COVID-19 patients around the world, and we took advantage of literature published by many, many different groups around the world. And at the end, we could identify two patterns, the green pattern and the red pattern. The green pattern is the Wuhan pattern. That means the prevalence of asthma is lower in COVID-19 than actually expected from the general asthma prevalent in the respective age groups. And you can see here this pattern can be observed in many countries. But when we have here the red countries, particularly North America, Australia, Britain, English-speaking countries mainly, where the prevalence of asthma among COVID-19 patients seems to be quite higher. And this, on the right-hand side, you see here the actually prevalence data in black among COVID-19, in white the asthma prevalence in the general age-matched population. The top bars represent the countries where we see the red pattern, and in the lower panel of this table, you see the countries in green, striking differences. So when we set out to build an intercontinental research network with colleagues in Stanford, 3 university hospitals in Germany, Moscow and Wuhan, and we had now the opportunity to assess these prevalences and also the comorbidities and other factors in this rather large, highly controlled cohort of about 5,000 inpatients, COVID-19 inpatients. And again, we confirmed the original observation, meaning that most countries show a lower prevalence of asthma, and the exception really is the US, with an inverse relationship, the prevalence of asthma among COVID-19, 18%, in the general population, 10.6%. So what could be the reason for this? Well, we need to realize that type 2 inflammation with Th2 cells, eosinophils, and mast cells carry many functional aspects which actually lower the risk for SARS-CoV-2 infection. And this includes the production of Th2 cytokines. Particularly IL-13 is downregulating the AC2 receptor expression on the epithelial layer, and this is the receptor mainly responsible for the invasion of the cells by the virus. But also eosinophils and mast cells carry important antiviral activities, and inhaled corticosteroids also contribute to downregulation of the ACE2 receptor. However, once an asthmatic person has acquired the SARS-CoV-2 virus, once the SARS-CoV-2 virus overcame this protection provided by type 2 inflammation, then this particular asthmatic individual is of higher risk for severe development. And this is related to the fact that many other antiviral activities are downregulated, are suppressed in the presence of type 2 inflammation. And this includes the high antiviral activity provided by Th1 and TC1 cells, the production of interferon alpha by PDC cells and others. So on the one hand side, it is good to have a type 2 inflammation, which lowers the risk for infection, but if infection occurs, then the asthmatic patient is of higher risk for severe development. And this is also highly related to the presence or absence of comorbidities. If asthmatic patients have comorbidities, mainly related to metabolic dysfunctions like obesity, type 2 diabetes, hypertension, cardiovascular disease, then these asthmatic patients are not anymore protected through type 2 inflammation because they have already a chronic subclinical type 1 inflammatory response with increased subclinical levels of interleukin-6, with high activities of Th17 cells, of CD14 cells, higher expression levels of ACE2 receptors and others. So my message here is it depends really on the endotype of asthma or the endotype in the non-communicable diseases, whether the patient is of higher or of lower risk for the development of diseases. So in summary, SARS-CoV-2 cannot be considered a classical, a conventional respiratory virus like rhinovirus, influenza, and RSV. Influenza, rhinovirus, and RSV are causing severe asthma and COPD exacerbations, whereas this is not the case with the SARS-CoV-2 virus. And in case we have a patient with a classical dominant type 2 inflammation, when this patient is of lower risk for SARS-CoV-2 infection, for severe COVID-19, and also shows a reduced mortality rate. However, in the absence of type 2 inflammation, in the presence of non-type 2 inflammation in COPD patients, the opposite happens. This patient is of higher risk for severe and even lethal outcome in COVID-19. So in summary, the Wuhan cohort showed us that asthma is not a comorbidity of severe COVID-19. The global perspective shows us that we have a different pattern, a diverse pattern among different countries. Type 2 inflammation may protect for SARS-CoV-2 infection. SARS-CoV-2 is not a respiratory virus causing severe asthma exacerbation, and SARS-CoV-2 has a very unique interrelationship with the host-virus interaction is different from other respiratory viruses. So with that, I would like to thank all of our collaborators in Marburg and around the world. And I would like to thank our funding bodies for continued support, and I would like to thank you for your attention. Thank you very much.
Hi, this is Jenny Ting, and I just want to thank the organizers for inviting me to give this talk on microbiome and innate immunity. Due to the time zone difference, I will not be able to be there to answer questions, and I just want to thank you for inviting me again. So as all of you know, there are a lot of innate immune receptors that have been discovered, and this is just a diagram of the different ones, and the ones that we will focus on today is really this family. They're known as NBDLR containing proteins, also known as the NOD-like receptors. They all have a nucleotide binding domain followed by a leucine-rich repeat. And we came upon this family about 20 years ago by looking for proteins with NBD and LR domains because we were studying one of these proteins called C2TA. And we came upon this family and realized it's a really large family of 20-some members in humans and 30-some members in mice. At the time, several of these were known already, but we found another about 16 of these genes. The most famous subfamily of this large family are the inflammasome-inducing NLRs, and then there are several proteins that also downregulate immune signaling, such as NF-κB MAP kinases or interferon, and I will start by focusing on one of these called NLRP12. Now, there are lots of work done on looking at the role of these NLR proteins in gut microbiota, and I just want to refer you to a paper that we published in JEM in 2020 by Hal Guo, Sarah Gibson, and myself, and that summarized many of these discoveries. Now, like I said, we will focus on one of these proteins, NLRP12. This is an intracellular non-like receptors. Actually, all of them are intracellular, but most of them, we don't know what their ligands are. There's a few that we know the ligands, but most of them, we don't know what the ligands are. And NLRP12 is one that inhibits NIC kinases, which is in the non-canonical NF-kappa B pathway. So this blocks NIC kinase activation, therefore reducing IKK alpha, therefore reducing the non-canonical NF-kappa B pathway. It also crosstalks with the canonical NF-kappa B pathway by blocking NF-kappa B activation as well. So mostly we're focusing on this part, but in the literature, other papers have also shown it reduces this part. Finally, NLRP12 has been also found to be important in an inflammasome situation, so it could serve multiple roles. And here it just shows some of our studies linking the role of NLRs with colitis. And the reason we were interested in exploring this is because one of the best genetic linkage of NLRs to Crohn's disease is NOD2. And so we wanted to know if the other family members also have a role in colitis. And so just to show you the biochemical changes, this is NLRP12 dendritic cells. This is from wild-type dendritic cells, and these are activating with different stimulus. In the wild-type, you don't see P100 converting to P52, and this is a typical conversion for non-canonical NF-kappa B. But if you look at NLRP12, it actually has a conversion. What this suggests is NLRP12 is usually there to block this pathway. If you remove this gene now, the NIC kinases are activated and P100 is processed to P52. If we put this in a DSS colitis model, now this is a typical model for both gut leakage as well as for colitis. What we observe is the wild-type mice do relatively well in this system, but the NLRP12 knockouts do terribly. A lot of them lose their survivability. And if you move this on to an AOM DSS model, which is a model for colon cancer, you see lots of polyps in the NLRP12 knockout mice, again suggesting this gene not only blocks inflammation here, but also blocks colon polyp formation. And if you look at NIC activation in these tissues, you will see that NIC is highly activated in NLRP12 knockout, and NIC-regulated chemokines indicated here are also increased. So this is our paper, and this is a paper by Zaki and colleagues from the Kaneganti lab. So years later, we had a new student, and he came and repeated these experiments. And this is, again, the same experiment looking at NLRP12 knockout mice. These mice lose more weight, so they have more severe symptoms. And if you look at survivability, again, the NLRP12 knockout mice lost survival ability. However, if we put these in a germ-free facility, if you look at NLRP12 knockout and wild type, there actually is no difference between the two. So this was really surprising to us. And if you look at survival rates, there's no difference. So this suggests that the genotype and phenotype connection here is mostly due to microbiome, because you don't see this in germ-free facility, but you see this phenomenon in specific pathogen-free facility. And this paper was published 4 years ago now in Nature Immunology. And if we look at the microbiome by doing 16S profiling, NLRP12 knockouts have less diversity based on the observed species numbers. And in addition, if you look at UniFrac of coordination plot, you see that wild type and NLRP12 have very different microbiome makeup. And this is indeed shown also in the dissimilarity value here. So we wanted to know specifically what is different between NLRP12 knockout mice and the wild type. So what we did is we actually kept these mice in 2 different vivaria. And the reason for this is in some studies, it's only specific for one housing component. So that wouldn't be interesting because it varies by housing. So we made sure that whatever we found was found in 2 different housing environments. And in addition, we made lots of litter maids to show that this is not due to effects such as different caging, different mothers, and etc. So we used litter maid controls. And what we found is NLRP12 knockout in red had reduced Bacteroides, they have reduced Clostridiales, and they have reduced lactose-free ECA. So these are the a very highly reproducible drop in species in NLRP12 knockout mice. Interestingly, these are the same groups of bacteria that are also lost in IBD patient published by the MIT group. So this suggests that these loss of these specific bacteria may have relevance in human diseases as well. So what we did is we did a number of studies, including a transfer study, where we transfer lactose-free CA into NLRP12 knockout mice and see if it reduced both colitis as well as inflammatory cytokines. And that is indeed what we saw. So this is, again, the DSS model where we fed the mice with lactose-free CA, then gave them DSS, and we followed their weight loss. And NLRP12 knockout, again, lost a lot of weight because of severity of colitis. If we fed these mice with lactose-free CA, they did better than even wild type. So this is how well they did. The wild type mice that's unfed with bacteria is down here, and bacteria cause a little bit of increase in weight. So lactose-free CA is a good bacteria that was able to reverse the colitis phenotype in NLRP12 knockout mice. At the same time, we also looked at inflammatory cytokines. So these are the different cytokines that we looked at. And the first group is wild type with just bacterial media. The second group is those fed lactose-free CA. And to be honest, in the wild type, we don't see much of a difference between those that are fed bacteria media and the bacteria. But in NLRP12 knockout mice, we saw a big difference. This is NLRP12 knockout controls. They produce significant amounts of IL-1 beta, IL-6, IL-12, IL-12, and TNF. And if we fed NLRP12 knockout with lactose-free CA, we see a drop in all of these inflammatory cytokines. So this was really interesting, suggesting that this bacteria not only reduced colitis, but colitis-associated cytokines as well. Now, these poor mice, in addition to having more severity in colitis, it also, if you put these mice on a high-fat diet indicated here, or even when they're on a low-fat diet, their fat component, the GWAS, and inguinal fat adipose tissue, you can see are bigger than the wild type. So they have more fat. And this is measured here in NLRP12 knockout mice, either on a low-fat diet or high-fat diet, have increased amounts of gut-associated adipose tissue. And in addition to that, they also have increased glucose, suggesting that they actually may have a risk factor for type 2 diabetes. And this paper was published 2 years ago. And in addition to that, we also found in this model that we fed these mice high-fat diet, we have increased TNF. NLRP12 knockouts have increased TNF. Wild type have increased TNF when comparing low-fat diet and high-fat diet, but the NLRP12 knockouts have even more increased enhancement of TNF. And this is also true of IL-6. Now, it has been historic that these are inflammatory cytokines that have been linked to obesity. So then we wanted to know, what if we put the NLRP12 knockout mice and wild-type mice in the germ-free facilities where there's no bacteria? And what we saw is as the following. So this is NLRP12 knockout mice in the SPF condition. So these are your normal housing conditions. So they gain weight compared to your wild-type mice. However, if all of these are both of these are put in the germ-free facility, there's no difference between the two, suggesting that once you take away the microbiome, NLRP12 and wild type do not show any change in weight gain. And if we look at IL-6 and TNF, the two key cytokines that have been linked to obesity, again, we see the same thing. This is really clear here of TNF. If you look at wild-type mice, they have less TNF than NLRP12 knockout. If you put all of these on antibiotics, the TNF level goes down. If you put these in germ-free facility, TNF levels are equivalent. There's no difference between these two. Again, suggesting it's the microbiome that's causing this difference in cytokines that are related to adipose tissue. So finally, we asked the question, what if we just fed these mice with the bacteria? Would they be able to lose weight? Would they be able to maintain normal weight instead of gaining weight on a low-fat or high-fat diet? And this is high-fat diet results shown here. This is wild type, and wild type fed with the lactose-free CA, the good bacteria. There's really not much of a difference. And the explanation for this is because the wild type has sufficient lactose-free CA. So even though you fed them, this bacteria is not going to make a difference. But if you look at NLRP12 knockout mice, there's a big difference. Mice that are fed with the bacteria lost quite a bit of weight, and their body fat is much reduced to about 10%. So this is almost like an athlete. But these mice didn't really train for anything. They just took in this bacteria. So this is pretty remarkable in showing that when you have a high-fat diet, you induce inflammation. It can cause all these inflammatory cytokines to be produced. And I skipped quite a few steps, but what we found is that with these cytokines, you see increase of certain bacteria that are associated with obesity, this bacteria, but you see reduction of bacteria such as Lactobacillus, which are good. But when you fed mice these good bacteria, it can actually block the high-fat diet and induce inflammation. And NLRP12 is there to block this hyper-inflammatory state and block dysbiosis. So this is a good gene that promotes good bacteria to fight against both colitis as well as inflammation. Now I just want to share with you our latest finding on radiation and microbiome. And this is just to show you that in radiation, there are good radiations, for example, for cancer therapy, but there's also worries about a nuclear accident or a dirty bomb, for example, for a terrorist. So in all of these cases, what you're most worried about is, first of all, you have a hematopoietic syndrome where the bone marrow is destroyed. Then you have a GI syndrome where the GI start to have all sorts of problems in terms of leakage, dysfunction, death, etc. So a few years ago, Hao Guo in my lab was looking at radiation-induced damage, and she found upon radiation, there's a small percent of the mice that actually survive radiation. And this is compared to normal aging process where mice can survive aging, but about 2 years of age, they start to die. But these mice, which are survive radiation, they live a pretty normal lifespan, and they start to die about 2 years of age as well. So we wanted to know if this has anything to do with the microbiome. So what we did is we profiled our microbiome, those of these ones that are survived radiation and live for a long time, which we call the elite survivors, and controls, which just age naturally. And we found that their microbiota makeup is quite different. And if you look at these two, this is specific bacteria, they also look very different. So then we want to know if we can do a fecal transplant study using the fecal material from these elite survivors versus age-matched control and see if they can protect recipients from radiation-induced death. So we took the fecal material from control mice, which are just old mice, or elite survivors, which are old mice that were radiated but survived it. And we took these fecal material, transferred them into germ-free mice or into SPF mice, and the results are quite similar. And I'm just presenting the germ-free mice because that's the much cleaner model. And what was remarkable is that these germ-free mice, once they received microbiota from elite survivors, they did really well and were able to survive a radiation exposure that was sublethal or close to lethal. So this was pretty remarkable. And in addition to that, the clinical score of these mice, the red mice, were really low. So they didn't have much of a severe clinical outcome, suggesting that whatever bacteria is in the elite survivor was able to protect the recipients. So we took these recipients and profiled their microbiome, and their microbiome from recipients of elite survivors versus control are quite different. And if we look at the specific bacteria, we found a number of bacteria are increased in recipient fecal material that received elite survival fecal material. So this is really a transfer from the elite survivors. And so we took 4 of these bacteria that are enhanced in the elite survivor recipients, and we transferred these and looked to see if they can increase survival to radiation exposure. And this is Lactobacillus, Enterococcus faecaceae, Lactobacillus, and Bacteroides. And what we found is we treated mice with these different bacteria separately and then radiated them. And what we found was the Lactobacillus was the best in protecting these mice from radiation exposure. And Enterococcus did pretty well, and Lactobacillus did pretty well, but this is similar to a previous study that has shown the effect of Lactobacillus in radiation protection. And so we wanted to see if the hematopoietic syndrome is being reduced, and it was. So if you look at non-radiated mice and BHI, there's lots of holes here in the BHI treated, but radiated. Then this is non-radiated controls. You see lots of holes in the bone marrow, but if these are treated with Lactobacillus, you start to see purple activities. The cells are proliferating, they're starting to come back. And if you look at the white pulp, they're tiny, and the ones that are radiated, and the ones that are radiated but receive bacteria, they look pretty good. And the same goes with the GI syndrome. So the colon, you see a big gap in those that are not treated with the bacteria, but those treated with lactose-free, they look pretty good. And the villi length is shrunken here, but the villi length is normal. So the lactose-free state was able to really protect both the hemopoietic syndrome and GI syndrome. And it also protected against gut leakage, and the radiation protection lasted as long as we measured it, which was 30 days. We also look at patient samples, and we were able to collaborate with Duke and Memorial Sloan Kettering. And we look at patients with diarrhea that lasted over 10 days after radiation, or with diarrhea that are better, so they lasted less than 10 days. And this better group all have higher amounts of lactose-free C, Enterococcus cacacea, and lactobacillus. So what this says is the patient with less severe syndrome have increased amounts of these beneficial bacteria, similar to what we would predict from our previous data. Then in addition to that, one of the major metabolites downstream of lactose-free C is short-chain fatty acid. So we treated mice with short-chain fatty acid, and the results are also very good. So this is the different short-chain fatty acids. So this is propionate, butyrate, and acetate. So all three, especially propionate, was able to improve the survival rate after treating these mice with short-chain fatty acid and radiating them. So in addition, if we look at the bone marrow and spleen, again, those that are treated with propionate had a lot more activities going on in the bone marrow as well as in the spleen. So we also wanted to know if there are other metabolites that are important. Short-chain fatty acid has been shown by others, such as Previn Reddy, as being important in graft versus host disease and radiation exposure. So we wanted to see if we can find novel metabolites that can also help in radio protection. So we were able to recruit a collaborator, Kun Lu, who did metabolomics for us. And what you see is metabolomics profile of those that received elite survivor fecal material is very distinct from those that were just enriched in controls. And looking at these two, we found that this group, the group that survived, has tryptophan metabolites. So we took two different tryptophan metabolites, ICA and chitonurin, and we treated mice with these and then radiated these mice. And they actually also achieved increased survival rate compared to control mice. So this is over 300 days. So again, a radial protection that lasted for a long time. So just to conclude for this final part, so what we found is in the gut bacteria of mice, there are beneficial bacteria such as these. And if they have a lot of these beneficial bacteria, they can protect against radiation-induced loss of hemophilia as well as intestinal damage. And in addition, we found two different metabolites that are downstream of these bacteria, propionate and also tryptophan metabolites. So this shows the power of microbiome in mitigating disease syndromes. So I just want to thank the people who are involved in this study. So a lot of these people, Coy, Leo, Justin, Aga, have since left the lab. Coy is a faculty at Virginia Polytech and Justin at the University of Arizona. And in addition to them, I also want to acknowledge Hao Guo, who did the bulk of the study and the radiation study with help of Emily, as well as others in the lab. And we also collaborated extensively with Balfour Sarter, who helped us with the germ-free facility, Vince Young, who gave us the bacteria, and also with Duke and Cornell and Sloan Kettering for the human studies. And I just want to thank you so much for your patience, and I wish you have a wonderful meeting.
Now we are open to discussion. Even though we have some questions, we do not have enough time to discuss. So at first, Dr. Kwong, can you read the question and you can make answer?
Sure. So there was a question about the use of dilute bleach baths. There was a question whether or not the use of dilute bleach baths in pediatric patients does the concern of reduction or eradication of the skin microbiota have a negative outcome that we're not considering. And based on my calculations when we were originally recommending dilute bleach baths to patients, in that the parts per million of chlorine do not exceed what is normally found in a swimming pool. And so based on the relative safety of swimming pools, that there was encouragement to use this, particularly since we see good skin outcomes with dilute bleach baths. And it's not thought that the dilute bleach baths actually eradicate completely the skin microbiota, and especially with the appendages in skin, such as hair follicles and ducts, that there are microbes that are still preserved in those skin appendages that can also repopulate the skin, even if there is a reduction in microbes. So it is a fairly brief time point that there may be a reduction in microbes, but there is maybe another thing to think about with sodium hypochlorite is that there was a paper in JCI, I think it was 2013 by Thomas Leong and colleagues, where topical sodium hypochlorite actually reduced NF-kappa B mediated inflammation in skin in a mouse model. And so the sodium hypochlorite may actually be doing something beyond an antimicrobial effect, may actually have an anti-inflammatory effect as well. But that was not extensively studied in humans. So that's something to think about.
Okay, I have the very short question from your SRS study. You mentioned about the SRS from the AD flare, induced epithelial thickening and immune cell infiltration. Do you have some evidence is that direct microbiota effect or some other material, toxic material and the other things Can you explain that?
Oh, so that's that we did not look at whether or not there was a metabolite or a direct bacterial, but those are all very good questions. That is a good question about what exactly is eliciting the host response.
Okay, thank you for your answer. Now we are closing this interesting microbiome plenary section. Thank you very much for attending this plenary session and giving clear lectures for 3 speakers. Then I hope you will enjoy the whole APAACI program, the Congress. You can find lots of interesting sessions in this Congress. Take care of your health in this pandemic and have a good luck. Thank you again for your kind attention. I'm closing this session. Thank you very much.